Guys, welcome back to my channel. In today's video, let's continue the gene transfer method, that is conjugation. Conjugation was first time discovered by Joshua Lederberg and Edward Totham. Now let's see the definition of conjugation. We already know that conjugation is nothing but the gene transfer method in which DNA or whole bacterial chromosome or plasmids are transferred from one bacterial cell to another bacterial cell by direct cell to cell contact. Thus it was included under the horizontal gene transfer method. Now let's see which types of cells are involved in conjugation. So we already know that bacterial cells have bacterial chromosomes as well as F plasmid as extra replicating DNA molecule. Okay, so there most of the time F plasmids are involved in conjugation, and conjugation always most of the time done by F plasmids. So there are four types of cells present in conjugation process. First one is the F plus cell, that means it has bacterial chromosome as well as F plasmid inside it. Second is the F minus cell, in which it lacks F plasmid. Okay, it lacks F plasmid. Third one is the F prime cell. It means that during replication, the bacterial chromosomes gets detached and integrates inside the F plasmid. Thus, it makes it as F prime. That means these cells have, this plasmid have bacterial chromosome integrated into it. Okay, and HFR cells means what? F plasmid containing bacterial chromosome inside it. Okay, we will see one by one in detail during the explanation. So these types of cells are involved in conjugation. Let's see the general mechanism of conjugation. That means transfer of conjugative plasmids by conjugation in gram negative bacteria. We will see conjugation in gram negative bacteria. Conjugation can happen in gram positive as well as in gram negative bacteria. Let's see the conjugation in gram negative bacteria. So first one is the F plus conjugation, that means conjugation involving F plasmids. So in this conjugation, bacterial conjugation, in the first step, the donor F plus cell, uh, the donor F plus cell having F plasmid inside it. On this F plasmid there is TRA locus present, which contains pillin gene. Okay, F plasmid has TRA locus which contains pillin gene, which initiates the conjugation process and forms pili or sex pillars to initiate the conjugation process. So F plus cells produce sex pillars that extends toward the F minus cell. Okay, then in next step, uh, sex pillars connects F plus and F minus cells. That means conjugation has been initiated, and the relaxase gene will nix the DNA strand of F plasmid at specific ORIT position. That means at F plasmid is also double stranded DNA molecule, we already know that. So relaxase enzyme will cuts the DNA at 3 prime position, that means at the ORIT region, that means origin of replication region. And then by using rolling circle replication mechanism, replication and transfer of F plasmid through sex pillars is occur. Okay, and in the last step, in the fourth step, donor, that means 5 prime to 3 prime DNA transfer has been done. Then donor cell also have F plus plasmid, and recipient cell also have F plus plasmid. So in this way F plus conjugation is occurred. Now conjugation can also be done in high frequency recombinant plasmids. Okay, now what are HFR plasmids or high frequency recombinant plasmid? So these are the F plasmids only, but during the integration with chromosomal DNA, a F factor you can see in this diagram, F factor integrates with chromosomal DNA. That means you can see F factor integrates into the chromosomal DNA, thus producing HFR cell. Okay, and similar to the F plus conjugation takes place. That means this HFR cell will produce pili gene, then the strand will bridge will created between two cells, that means donor cell having HFRs and recipient cell which lacks the F plasmid. Okay, then by rolling circle mechanism, plasmid get transferred from one cell to another cell by through F prime to 3 prime direction replication is done, and in this way HFR high frequency recombinant conjugation takes place. And if you see, the frequency of recombination between HFR cell and F minus cell is high, but the frequency of whole F factor is low, because we know HFR is nothing but the small amount of F factor is integrated into the host chromosomal DNA. Okay, thus the frequency of transfer of whole F factor is low, and in this conjugation chromosomal DNA is always almost transfer, because you can see in this diagram all chromosomal DNA is getting transferred into the recipient cell, but not F factor. That's why recombination frequency of transfer of chromosomal DNA is high, but F factor is low. Now third is the F prime conjugation. Now F prime cell is what we know, HFR. Now F prime plasmids are what, similar to HFR but opposite, vice versa you can say, in which F plasmid integrates chromosomal DNA inside it. In HFR what we saw, chromosomal DNA integrates some sort of F factor, but in F prime, F plasmid integrates some sort of chromosomal DNA inside it. Thus F prime cells get formed. Okay, that you can see here, bacteria which contains F factor and a part of chromosomal DNA integrated in it is known as F prime bacteria, or you can say F prime cells are formed from HFR cells. That means HFR cells having this bacterial chromosome, and this short part is F factor. Okay, this is HFR bacterial chromosome. Okay, so this is the bacterial chromosome and this is the short part F factor. Okay, and after the integration this F factor will integrate some sort of bacterial DNA, thus gets produced F prime cell. Okay, so F prime cells are formed from HFR cells during induction of F factor from chromosomal DNA. That means F factor is getting induced. Okay, in which F factor carries a portion of chromosomal DNA along with it. That means some sort of chromosomal DNA is carried out with F factor. In this type of conjugation between F prime cells and F minus cells, frequency of recombination is high, as well as frequency of transfer of whole F factor is also high. But we saw that frequency of recombination in HFR cell is high, but whole factor does not get transferred. But you can see whole factor, whole F plasmid has been transferred in F prime conjugation also. That means F plus conjugation and F prime conjugation transfer whole F factor. Okay, so these are the three types of conjugation. Now let's see the examples of conjugation. Most conjugation is mostly preferred gene transfer method by bacteria. We already know that. So this conjugation method has wide range of applications in genetic engineering and biotechnology, because this conjugation method can transfer 1000 to 1 lakh of base pairs of DNA from one donor cell to another donor cell. So the first example is Agrobacterium tumefaciens, in which it has crown gall tumor. Okay, but it can cause crown gall tumor in plants by transferring tDNA element present inside its plasmid, that means in Ti plasmid present in this agrobacterium tumefaciens bacterium, which can transfer to plant cell where T elements become incorporated into the plant cell genome and thus can cause crown gall tumor in plants. Second one is conjugative plasmids encoding antimicrobial resistance genes are called R plasmids. Okay, that means we have to induce antibiotic resistance in some other bacterium. So one bacterium having antibiotic resistance gene inside its plasmid can be transferred to another bacterium to induce antibiotic resistance. So here mentioned Shigella species have antibiotic resistance inside it, which can induce antibiotic resistance to other bacterium to resist to out in to result in a widespread outbreak of antibiotic resistant Shigella mediated dysentery. So these are the examples of conjugation. We have already saw transformation and transduction methods of gene transfer in detail. The complete video has been uploaded on my YouTube channel and the link will be provided in the description box. So do check out that videos and like, share and subscribe to my channel. Thanks for watching.